Students, let me know in the chat if I am audible right now. Quickly let me know in the chat if I am audible and visible. Now, check now. Students, as I was saying, welcome to Vedantu NEET English. This is Baswaraj, so I'm your biology master teacher. So, what will we be doing in today's class? So, in the previous class, we finished all the starting part of the chapter, right? Starting part of the chapter we did in the first class. It could be starting off with the basics of basics, just simple tissues, complex tissues, simple tissues such as parenchyma, collenchyma, sclerenchyma, complex tissues such as xylem and phloem, meristematic tissues, primary meristematic, secondary meristematic, all of that is done. Now, in today's class, what is going to be our goal? In today's class, we'll be doing the most important part of the chapter. It is the sections. It is the sectioning. So let me know in the chat how many of you are scared of such beautiful sections. Tell me in the chat how many of you are scared, because it can become overwhelming, right? And I have always told you the way to approach the anatomy is by diagrams. You cannot by heart any of it. You need to remember the diagram, and from the diagram you need to make your notes. You need to make your notes from the diagram itself, right? So we'll be doing the similar way. It could be starting off with dicot and monocot root, dicot and monocot stem, dicot and monocot leaf, all the following different types of sectioning in today's class. So let me know in the chat if you are excited for today's class, because we'll be starting the class with a simple question. See here, this right here is a very simple question. You might find it a little difficult to process it right now, but trust me, by the end of today's class, you will be able to solve such questions with so easily you'll be able to solve such questions. So quickly, green hearts in the chat, because we'll be starting the class with something called as tissue system. We'll be starting the class with something called as tissue system. Now students, now I'll be making you write the notes. I want you to write the notes with me. So you will have the copy of notes with you. But if you want a notes for yourself, which I write on the board, that will be available on Baswaraj Sir Biology and Vedantu NEET English Telegram channel. The notes will be uploaded there. Okay. So let's start the class with something called as tissue system. Tissue system. Students, you need to understand, under tissue system, there are 3 different layers. There are how many layers? There are 3 different layers. The first layer is called as epidermis. It's called as epidermis. The second layer is called as ground tissue. It's called as ground tissue. And the third layer, and the final layer, the third and the final layer is called as vascular tissues. Vascular tissues or vascular bundles. Vascular bundles. So all the 3, one by one, we'll be learning in today's class, and after that we'll be going to the different types of sectioning. Now, for example, when we think of the epidermis, this right here is the entire section of the epidermis. Now, this section of your entire epidermis, on the top, do we have some kind of epidermal appendages? Yes. On top of the epidermis, we can either have root hair, or some of you might have seen the tomatoes, right? Tomato plant. In the tomato plant, have you noticed the stem also has some type of hair? That is called as trichome. So under epidermal hair, there are 2 topics which we will be dealing. 2 topics will be dealing under epidermal hair. Then we have the arrangement of epidermal cells here, the epidermal cells. Now in the epidermal cells, we also have certain structure called as stomata. Also have a structure called as stomata. It's not visible here, so I'll write here. We will have a stomata here. We will have stomata. So all the 3 different epidermal appendages, the epidermal cells as well as stomata, will be covered under epidermis, only under epidermis. Okay. Now students, quickly let's start with the starting epidermal hair now. Ready? I want you to write down the notes one by one with me, so you will have a better understanding. First, can we start with root hair now? Root hair. The first thing you need to know is the origin of root hair. Where did the root hair even originates from? For example, if you know the morphology of flowering plants chapter, if you remember, we did a diagram of root. Yes, we did a diagram of root. We had the meristematic region, we had the region of elongation. Yes, we also had the region of maturation. Region of maturation. Now from this region of maturation, students, from this region of maturation, did we see a development of minute hair-like structure from the region of maturation? Yes. That is the actual origin of root hair. Actual origin of root hair. So tell me in the chat, is the root hair originating from inside or outside? Obviously it is originating from the outside. So can I tell root hair origin is exogenous in origin? Exogenous in origin? Yes, it is going to be exogenous in origin. Done? Yes, exogenous in origin. The same root hair right here, the same root hair, can I tell it is also unicellular? Yes, it is also unicellular in nature. The same root hair, what could be the function of root hair? Tell me in the chat. What could be the function of root hair? The main function of root hair is to increase the surface area. Yes, increase in surface area. Done. The three different points for root hair. Now let's go to trichome. Let's go to trichome now. What about trichome, students? What about trichome? What about trichome? Okay, let's understand trichome now. Now first of all, trichome, you need to understand the location. See how I told you, the root hair is on region of maturation. Your trichome technically develops. Trichomes technically develop on the stem. So can I tell, see on the stem, on the tomato stem, it will grow out like this. So can I tell it is also called as stem hair? Can I also call it stem hair in it? Yes. But unlike your root hair, your trichomes are technically multicellular in nature. They are multicellular in nature. It is the same trichome, students. It is the same trichome. What will be the function of trichome? The main function of trichome in the chat. The main function of trichome is to reduce the water loss. To reduce the water loss. Reduce the water loss. That is by reducing the respiration. Reducing the respiration. That is your trichomes. Now students, sometimes what happens is, this trichomes right here, right? They can either be soft or hard also. They can be soft or they can be hard. The trichomes can be soft or it can be hard. The same trichome can also become secretory in nature. It can release certain substances. It can release substances. It can release substances. Release substances. It can also be septate. That is, septa formation can also happen. It can be septate or aseptate. Aseptate. Both are possible here. Both are possible here. So first we learnt about root hair, that is exogenous in origin, unicellular, increasing the surface area. It is originating from region of maturation, that is root hair. Then we come to trichomes. Trichome is stem hair, multicellular, reduce the water loss, soft or hard, septate or aseptate, release certain substances, that is, it is secretory in nature. It is secretory in nature. It can release certain substances. It can release certain substances. That is trichomes. Done. This is basically your epidermal appendages. So if you go back, epidermal appendages done. Two types, root hair and trichome. Now can we learn some properties of epidermal cells now? Can we learn some properties of epidermal cells now? Yes. What are the main function of epidermal cells, friends? Now, you tell me in the chat. If you look at this diagram right here, if you look at the diagram of this outer layer, can you see this one? This outer layer right here. Can I tell first point, simple point? Let's go very simple, scratch basics. Can I tell it is the outermost layer? Yes. Very simple, right? It is the outermost layer. Can I tell? Yes or no in the chat? So can I tell it is the outermost layer? Outermost layer. What is the function of the epidermal layer? The main function of epidermal layer is protection. Main function is going to be protection. Yes, main function is protection. Now, if you look at this particular epidermal layer, is it only a single layer or is it multiple layers? Obviously, can you see? It is made up of a single layer. Yes. So can I write it is made up of single layer? Yes, it is made up of a single layer. Now what is it made up of? Which type of cell? Parenchyma, collenchyma, sclerenchyma. So you should know by this by now, students. Most of the epidermis is made up of parenchyma. Most of the epidermis is made up of parenchyma. Parenchyma. Now look at the shape of the cells. Look at the shape of the cells. If I draw it somewhat like this, look at the shape of the cell. Can I tell they are like barrel shaped cells? Can I tell? Do they look like barrel shaped cells? Yes or no in the chat? Yes, they look like barrel shaped cells. Barrel shaped cells. Yes, barrel shaped cells. Yes, barrel shaped cells. That is your entire layer of epidermis. Now, just look at the diagram and understand. Do you find any type of intercellular layer anywhere? Intercellular space anywhere? Do you find any space between the two cells? Obviously not. Obviously not. So can I tell intercellular space is absent? Yes, intercellular space is absent here. I can tell that. Right. Now, next thing I want you to understand here is, if intercellular space is not here, what about the chloroplast? What about the chloroplast? I know it is parenchyma. It is not always compulsory that parenchyma should have chlorophyll or chloroplast. So students, remember this point very clearly, because you can get a lot of questions from here is, chloroplast is actually absent here. Chloroplast is absent here. Absent here. Except at the guard cells. Except at the guard cells, you mostly find the chloroplast. Except at the guard cells, you will not find any type of chloroplast. So chloroplast is completely absent. You do not find chloroplast in the epidermal cells except at the guard cells. Done. Except at the guard cells. Now, epidermal cells in the case of stem and leaf, will they have a thick waxy coating on top? Will they have thick waxy coating on top to reduce the transpiration? Yes. That waxy coating is called as, tell me. The waxy coating on the top of the epidermal cells is called as cuticle. It is called as cuticle. Cuticle is present. Cuticle is present. Cuticle is present here. Yes, cuticle is present. Now students, this cuticle, do you find it in the roots? Do you find it in the roots? No. So that's point which you write down. Cuticle is absent in the case of root. It is absent in roots. Absent in roots. Now let's look at all the different points here. If you do not understand any point, ask me. Epidermal cells is the outermost layer. Yes sir, outermost layer. Main function is protection. It is a single layer made up of parenchyma. And in this parenchyma cells, there is no chloroplast except at the guard cells. Intercellular space is completely absent. They are barrel shaped cells. Cuticle is present on the top, but cuticle is absent in the case of roots. Cuticle is absent in the case of roots. Do you realize, do you need to mug up all this? No. If you look at the diagram, all of these following points should come to your mind. It's anatomy. Anatomy, if you by heart, you will never progress further. Anatomy is supposed to learn with the help of diagrams. See, beautiful. With the help of a single diagram, we were able to write down so many points. Done. Can we go to the next slide now? This is the epidermis. Now students, I told you at a specific locations in the epidermis, there is chloroplast is present. That present part is called as stomata. It is called as stomata. Now let's draw stomata quickly. Let's draw the stomata. Yes, this is the stomata right here. This is the stomata right here. Now students, here this round structures you see here, this two structures, what do you think are called as? What do you think they're called as? Here, these two structures right here, they will mediate the opening and closing of the stomatal pore. Opening and closing of stomatal pore. Now these two cells are called as guard cells. They're called as guard cells. Now around the guard cells, there are some type of special epidermal cells. There are some type of special epidermal cells here. Around the guard cells. You find them around the guard cells, some type of special type of epidermal cells. So yeah, some type of epidermal cells. What do you think they're called as? The special type of epidermal cells around the guard cells are called as subsidiary cells. They're called as what? Subsidiary cells. Okay? They're called as subsidiary cells. Around this, later on we will have epidermal cells here. This is external to the subsidiary cells is normal epidermal cells, which we just learned. Normal epidermal cells. So let's understand one by one now. Let's understand. We have the stomatal opening or pore. Then we have guard cells around it. Now around the guard cells we have the subsidiary cells. Around subsidiary cells we have the epidermal cells. Any doubt, quickly ask me. Any doubt, quickly ask me. This is the entire structure of stomata. See here. Now this structure of stomata can vary. For example, in the case of monocot, it is dumbbell shaped. Can you see? It's almost dumbbell shaped here. In the case of monocot, do you see it's almost like a dumbbell? Here, dumbbell shaped cells. Now what about in the case of dicot? In the case of dicot, it is bean shaped. It is bean shaped cells. Bean shaped cells. In the case of dicots. Now here, one more thing I want everyone to understand here is the guard cells plus stomatal pore plus the subsidiary cells. Listen to me very carefully. The guard cells, the stomatal pore or the opening and the subsidiary cells together, they are called as stomatal apparatus. They are called as what? Stomatal apparatus. So in the examination, if they ask you what is stomatal apparatus, you should be able to tell, sir, it is guard cells plus stomatal opening plus the subsidiary cells. All three together are called as stomatal aperture. They're called as stomatal aperture. Clear? Apparatus, sorry, stomatal apparatus. Stomatal apparatus. Not aperture. Apparatus. Done. That was the epidermal tissue. Okay. Now students, with that, your epidermis is over. Any doubt in epidermis, quickly ask me in the chat. Any doubt in the epidermis, quickly ask me in the chat. I believe there should be no doubt regarding epidermis. Now can we start with the ground tissue? One by one, is very simple students. Ground tissue is mainly made up of 4 different categories. We have the, first of all, what is ground tissue? All tissues except epidermis and vascular bundles. All the tissues in the particular sectioning apart from the epidermis and vascular bundles is a part of ground tissue. It is a part of ground tissue. The first part of ground tissue is nothing but cortex. Cortex. Then we have pericycle. Then we have pericycle. Pericycle. Then we have pith. Then we have pith. They are separate. All of them are separate. Pith. And finally we have the medullary rays. Medullary rays. The same medullary rays which undergo dedifferentiation. Dedifferentiation to produce interfascicular cambium. The same medullary rays which undergoes dedifferentiation to produce interfascicular cambium. Now, but our main focus should right now here be on the cortex. Now tell me in the chat, what is cortex made up of? What is the first part of the cortex? Now we have epidermis on top, right? We have epidermis on top. Just below the epidermis, there is something called as hypodermis. The first part of the cortex, students, the first part of the cortex is called as hypodermis. It's called as what? Hypodermis. First part below cortex is called as hypodermis. Now below hypodermis, what do we have? Below hypodermis we have the general cortex. We have the general cortex. General cortex. Now if I ask you, which is the last layer? Which is the last layer of the cortex? It's called as endodermis. It's called as what? Endodermis. Endodermis, remember students, endodermis is a part of cortex. Endodermis is a part of cortex. Please remember that throughout your life. Endodermis is a part of cortex. Hypodermis, general cortex, and endodermis. Endodermis. Is that point clear to all of you? Is that point clear to all of you? Yes. That is basically your ground tissue system. Ground tissue system. Now students, can we start with the vascular tissue system now? What is this vascular tissue system? It might seem complicated to you in the starting, but I'll make it easy for you now. I'll make it easy for you. Okay. Now students, listen to me very carefully. The entire vascular tissue system is mainly divided into two categories. Mainly divided into just two categories. Okay. Just two categories. Okay. What are the two categories, students? The first category is very simple. First category is we have xylem, phloem, xylem, phloem, xylem, phloem, xylem, phloem, and again xylem and phloem. Now students, in the first category, if xylem and phloem are in their own radii, can you see? They're present in their own radii. Xylem has its own radii, phloem has its own radii. Xylem has its own radii, phloem has its own radii. Where a vascular tissue is present in its own radii and in an alternating manner, that is called as radial arrangement. That is called, students, radial arrangement. Radial arrangement. That is called as radial arrangement. And this radial arrangement is mostly found in the case of roots, or only found in the case of roots. Okay. Now students, the second one is very simple. The second one is very, very simple. If xylem and phloem, up, one second, let me draw it. This looks like a rice cake. If xylem and phloem are present on the same radii, see here what happened? Xylem has a different radii, phloem has a different radii. Xylem has a different radii, phloem has a different radii. But in the case of the next one, xylem and phloem will be present on the same radii, not different. They are present together. Xylem and phloem are present together on the same radii. Such type of arrangement is called as conjoint arrangement. It's called as conjoint arrangement, and it is mostly found in the case of stem and leaf. Stem and leaf. It's mostly found in the case of stem and leaf. That is conjoint. Done. Done. Here same radii. Here same radii. Here different radii. Done. Now students, this conjoint can be further classified into two categories. Two different categories. Okay. Listen to me very carefully. Listen to me very carefully. Now here what happens is sometimes what happens is, this is the center part. Here what happens is, listen to me very carefully, all of you. Very, very crucial, very crucial. Now this is the center pith. This is the center pith. If xylem is present towards the pith and the phloem is present towards periphery, xylem inside, phloem outside, xylem inside, phloem outside, xylem inside, phloem outside, xylem inside again, and phloem towards the outside. Such type of arrangement where the xylem is present towards the pith, is called as collateral arrangement. Collateral arrangement. That is called as collateral arrangement. Okay. Now what is this? The other one, what am I talking about here? This is called as bicollateral. This is called as bicollateral, which is not given in your NCERT, but you should know this. Not given in NCERT, but you should know this. What happens in bicollateral, students? In the case of bicollateral, it's very simple. In the case of bicollateral, we have phloem. Once again, let me use a different color. We have phloem, xylem, phloem. Phloem, xylem, phloem. Phloem, xylem, phloem. Phloem and xylem and phloem. This is called as bicollateral arrangement. It is called as bicollateral arrangement. Phloem, xylem, phloem. Phloem, xylem, phloem. Such arrangement is called as bicollateral. But in your NCERT, we are only dealing with collateral arrangement. What is collateral, students? Where the xylem is present towards the pith and phloem towards the periphery. Is that point clear to all of you? Is that point clear to all of you? Tell me in the chat. Is that point clear to all of you? What is the meaning of conjoint, collateral and radial arrangement of the vascular bundles? Okay. Now, students, again we have two types under collateral. Collateral has two types. Collateral itself has two types now. Okay. What are the two types in collateral, students? What are the two types in collateral now? Okay. Listen very carefully. What happens in collateral? Okay. Now in collateral, what happens is sometimes what happens. Let me draw this. Actually, we'll just erase this. It is not exactly not collateral, but I'll show you. Okay. We have xylem and phloem. Xylem and phloem. Now, students, between the xylem and phloem, now, if between the xylem and phloem there is a separate layer like this, can you see? This separate layer is called as cambium. It's called as cambium. If cambium is present between xylem and phloem, I'll write vascular cambium, you will get confused. If vascular cambium is present between xylem and phloem, such type of arrangement is called as open arrangement. It's called as open arrangement. Now, if between xylem and phloem there is no cambium, there will be a line, but obviously make it dark green. Okay. Now, if between xylem and phloem, if there is no cambium, there is no vascular cambium, such arrangement is called as closed arrangement. It's called as closed arrangement. Done. All following points clear to all of you? First is called as radial arrangement, where xylem and phloem are in separate radii, most often in the case of roots. Then we have conjoint, present in stem and leaf, where xylem and phloem are present in the same radii. Can you see? Same radii. Then we have collateral. In collateral, xylem is towards the pith, phloem towards the outside. In bicollateral, phloem, xylem, phloem. Now, between the xylem and phloem, if there is vascular cambium, then it is called as open type. Now, if there is no vascular cambium between xylem and phloem, it is called as closed type. And students, remember, this open type is present in the case of stem. This closed type is found in the case of leaf, generally. In the case of leaf. If there is no, and even in the case of monocots we have closed. In the case of dicots we have open again. Dicots open, stem especially, stem. Now, what is the use of vascular cambium? The main use of vascular cambium is it is involved in secondary growth. So, if cambium is present, it means that a plant will show secondary growth. If the cambium is absent, it will not show secondary growth, just like a leaf. Can you think a leaf will show secondary growth? Do we have woody leaves? No. A leaf will never show secondary growth. Only a stem will show a secondary growth. Is that point clear to all of you? A leaf will not show secondary growth. That is why it says closed. That's why it says as closed. Now let's just quickly read NCERT lines if you have covered everything. Yes, we covered about tissue system here, epidermal tissue system, the ground as well as vascular tissue system. Yes, under epidermal tissue system, did we learn about epidermis? Yes, we learned about epidermis. That is elongated, compactly, yes, arranged in the form of a continuous layer. Yes, it is continuous layer. Single layer, made up of parenchyma, right? Large vacuole, I think I forgot to mention large vacuole, right on the large vacuole. Then it will have a cuticle layer. Yes, cuticle is absent in the case of roots. Then we have stomata. Yes, stomata we did. Epidermis of leaf. What is the function of stomata? To exchange the gases. Done. It has guard cells around it. Yes, guard cells are dumbbell shaped in the case of your monocots and bean shaped in the case of dicot. Yes. The outer wall of the guard cell, students, very important line. The outer wall of guard cell away from the stomatal pore are thin. For example, see here. Here. Can you see? Away from the pore, these ones are thin. They are very thin. The inner ones, can you see the inner one? The inner ones are thick. Why? Because the inner ones has to maintain the pressure. The pressure will be there if obviously water gas exchange is happening. So the inner walls are thick and the outer walls are thin. Outer walls are thin. Yes. Then there is subsidiary cells, and we learned about stomatal apparatus. Stomatal apparatus consists of your opening, stomatal opening, guard cells, subsidiary cells. Done. Every single point cleared. This is your monocot, that is dicot, dumbbell shaped in the case of monocot. Yes. Did we cover this? Yes. We covered root hair and trichomes. Root hair and trichomes. Yes. They can be soft or stiff, that's hard I told you. Branched or unbranched. Multicellular in nature, trichomes. Prevent water loss, the function. The ground tissue system, yes, consists of medullary, pith, pericycle and cortex. We wrote that. Yes. Ground tissue system consists of thin-walled chloroplast containing cells called as mesophyll cells, which are in the case of leaf. In the case of leaf. Now here we discuss entire radial arrangement. Yes. This is conjoint arrangement, xylem and phloem together. This is called as open. Why? It's called as open because KMBM is present here. Can you see? KMBM is present. Radial, conjoint, vascular bundles, closed. All the points covered, students. All the points covered one by one. Now, can you solve some questions? Let's solve some questions now. See this question and try to answer. I'll drink some water. The time starts now. Go. Time starts now. Go. In the figure given, which component has thin outer wall and highly thickened inner wall? So, which of the following will have? See, first of all, tell me A is the epidermal cells, B is going to be subsidiary cells, C is going to be guard cells, and D is going to be stomatal aperture, stomatal opening. Which particular cell will have thin and thick walls? Yes, Ravindrani. Which of the following will have thin and thick wall? Obviously, students, we learned, right? Thin wall and thick wall is present with the guard cells, right? So tell me, which one is guard cell here? Which of the following is a guard cell? Technically, tell me, show. This was a controversial question, remember? This was the controversial question. Students, remember, B technically here, it is pointing towards the subsidiary cells. C is pointing towards the guard cell. C is pointing towards the guard cell. D aperture. So, guard cell is the answer. Guard cell is the answer. Yes, guard cell is the answer. Did you understand? This was a controversial question, but there is nothing controversy about it. It is just how you look at it. If you know the different types of markings. Now, stomata in grasses leaves are. How is the stomata in grasses leaves? Tell me in the chat now. In the case of grasses, obviously it's monocot, and in the case of monocot, the shape of the guard cell is going to be dumbbell shaped. Stomata is going to be dumbbell shaped. Monocot is dumbbell shaped. Next question. The cortex is the region found between. What is cortex? Cortex is between epidermis and vascular bundles. Easy question, we did this. Epidermis and stele. It's endodermis, that's why you can't use it. It's supposed to be epidermis and stele. Now, what is stele? I'll tell you later on. Stele is basically made, I'll tell it off now only. Stele is basically made up of vascular bundles, pith and pericycle. All three together, it is the stele. We'll discuss that later on. Okay. Next question. Next question. I think some of you went wrong here, right? Cortex is between epidermis, the outer layer, and the stele here. The stele. What is stele made up of? Stele is made up of what? Let me write it here, otherwise you'll forget later on. Stele is made of three things. Pericycle plus vascular bundles plus the pith. That is the stele. That is going to be your stele. Okay. See here, this is the epidermis. This is the epidermis. Anything between is called as cortex. Called as cortex. Done. Next question. Specialized epidermal cells surrounding the guard cells are called as. Specialized epidermal cells around the guard cells are called as subsidiary cells. They're called as subsidiary cells. Easy question. NEET 2016 level question. Students, let me tell you all the questions which I have in today's class is NEET PYQ, because I want you to know whatever you're learning in the class is actually applicable to you, right? Actually applicable to you. Okay. See this. Vascular bundles in monocots are considered closed because. Why do you think the vascular bundles are considered as closed? What is the meaning of closed, students? The meaning of closed is that between xylem and phloem there is no cambium, right? See the answer now. Bundle sheets surrounding the bundle sheets. Cambium is absent. Obviously is here. Answer is here. Answer is here. Cambium is absent. Easy question. NEET 2015 level question. Next question, students. Closed vascular bundles lack. What do they lack? They lack the cambium. Students, do you realize just from open close, you have concept, you have many questions. Open means present, close means absent. Next question. In land plants, the guard cells differ from other epidermal cells having. What is so different about guard cells? I told you when I was teaching you about epidermis. I told you, right? In the epidermis, all the cells lack chloroplast. All the cells lack chloroplast except at one point. That one point is called as what? The one point is called as guard cells. Guard cells. And they had chloroplast. Students, if you are able to solve all the questions till now, quickly smash the like button. And I forgot to tell you also. And all of you quickly mark your attendance. By smashing the like button, because I'm checking now. I'm checking every single day. Okay. So have you understood everything till now? Yes or no in the chat quickly? Because if you have understood, the anatomy chapter is very easy, like similar to this. Anatomy class is going to be similar to it. And we'll be going with root now, mostly dicot root, then monocot root. One by one we'll go over every single point. I want you to make your own notes from this. Okay, make your own notes from here. Make your own notes. So it will be helpful for you to understand also later on. Okay, make your own notes. Don't forget to make your own notes. Can we start now? Can we start, students? Hema, Meghana, Arpana, Ravindrani, Divya, Aturu. Yes. Madhumita. Can we start now? Let's start with the sectioning now. This is what the main part of the chapter is, and you will be getting a lot of questions from here. Trust me on that. And what actually happens is students try to by heart this part, right? You tend to by heart this because you have never learned this final part with the help of diagram. So I'll be teaching only with the help of diagram. all the information which I'll be writing today, everything will be derived from the diagram only. And in the examination, when they ask you a question, the diagram should come to your mind and all the points should just flow in. All the points should just flow in. Okay, let's start now. Green hearts in the chat if you're excited for this next bit, which is going to be very interesting. It's very interesting. This right here, we have a dicot root now. We have a dicot root. Now, one by one, we'll be writing points. So, I want everyone to follow very carefully now. Follow very carefully. Okay. Now, students, let's start with the first layer. First layer. Can you see the first layer? We'll go first, second, third. In a simple fashion, we'll go. In a simple fashion, we'll go. Okay. The outer layer. Can we start with the epidermis now? Let's start with epidermis. Epidermis. Now, point number one. Students, epidermis here has a separate name. Let me tell you first of all that. The epidermis right here in the case of dicot root has a separate name called as epiblemma. It is called as what? Epiblemma. It is called as epiblemma. Okay, done. Now on this epidermis, do you find any type of epidermal appendage? Do you see in the diagram? Do you find any type of epidermal appendage there? Yes, sir. We find root hair there. We find root hair. Yes, we find root hair. Now tell me, is the epidermis single layer or is it multi-layer? Tell me. Obviously, students, if you look at the diagram itself, you can tell it is single layer. It is single layer. It is a single layer which is made up of parenchyma. It is made up of parenchyma. Single layer made up of parenchyma. Now, students, if this is a root, right here, do you think roots require cuticle? Tell me honestly. Do you think roots will require cuticle? No. So can I tell cuticle is completely absent? Yes. Cuticle is said to be absent here. Yes, cuticle is absent here. Now, what about stomata? What about stomata here? Think logically. Think logically. In the roots, do you need stomata in the root system? In the root, do you need stomata? Obviously not. So can I tell in the roots, stomata is also absent? Yes, stomata is also absent. Now, students, if you look at carefully at the single layer, which is present on the top, single layer, do you find any type of space between the cells? Tell me, do you find any type of space between the cells here? Obviously not. So can I tell intercellular space is also absent here? Can I tell intercellular space is also absent? Done. Epidermis is done already. That's all. That's all. Just from the diagram and the logical thinking, we derived all the information regarding the epidermis. Tell me, was this difficult? Tell me, was it difficult? No. It's very easy. Similarly, we'll go to the next layer now. Now, next layer is what, students? Look at the diagram. I will not tell, you tell me now. What is the next layer called as? It's written here. The second layer is called as cortex. Yes, students. The next layer is called as cortex. Cortex. Now, tell me in the chat, if you look at the diagram, do you think the cortex right here is a single layer? Is it multi-layer? Obviously, students, if you look at the diagram right here, cortex is multiple layers. Yes, many layers. Many layer, or can I say multi-layer? Yes, it is multi-layer. It is also made up of parenchyma. It is also made up of parenchyma. Also made up of parenchyma. Now, look at the diagram. All of you, look at the diagram right here. Can you see a small gaps here? Here. Can you see small gaps here? Yes, you can see small gaps. If you have your NCERT, open your NCERT also. So you can see small gaps. What are the small gaps? They're called as intercellular spaces. Between the two cells, small space. So can I tell, here we have intercellular space is present, but it is very less or absent in many cases. Done. Cortex is done. That's all. All you need to do is look at the diagram and tell me what is present. That's all. Now students, if you remember very clearly, I told you, the innermost layer of cortex is called as what? Go in the chat. The innermost layer of cortex is called as what? If you're watching it recorded, students, I want to say it out loud. The innermost layer of the cortex is called as, here, can you see? This right here. The innermost layer of cortex is called as endodermis. Is the endodermis. Endodermis. Now students, if you look at the endodermis, tell me the first point. Endodermis, do you think it is a single layer? Can you see? Endodermis is said to be a single layer. See here? It is made up of a single layer here. Yes, single layer. Single layer right here. Now, if you look at the epidermis, do you find, if you look at the endodermis, sorry, do you find any type of intercellular space anywhere? No. So, can I tell intercellular space is completely absent again? Yes. Intercellular space is absent here? Yes. Now, next point. Let's look at the diagram again. Let's look at the diagram again. Okay. Now, all of you know endodermis is part of cortex. Yes, endodermis is part of cortex. That we all of us know. Endodermis is part of cortex. All of us know. Now, let's write down. Otherwise, you'll forget it off. It is part of cortex. Made up of parenchyma. Made up of parenchyma. Now, next thing I want you to understand is the shape here. The shape of the cells here. Now, endodermis has a special shape similar to the epidermis. The cell shape here is very peculiar. The cell shape, can you see? It looks something like this. The shape of the cells looks something like this. Here. The shape of the cells looks something like this. Such type of shape of the cell is called as barrel-shaped cells. Barrel-shaped cells. They are barrel-shaped cells. Now, the interesting thing to note here. The interesting thing to note here is that on this barrel-shaped cells, there is some form of deposition. Tangentially, like this, right? Tangentially and also radially somewhere. Tangentially, radially, there is some form of deposition. It's called as subarene deposition. It's called as subarene deposition. And in the end, on the barrel-shaped cells, when we have subarene depositions, ultimately it's known as Casparian strips. Casparian strips. Casparian strips on the barrel-shaped cells. Subarene deposition, barrel-shaped cells. Done. Now you might ask me, sir, what is the main function of endodermis, students? What is the main function of endodermis? You might ask me, sir, what is the main function of endodermis? The main function of endodermis is it acts as a checkpoint. Acts as a checkpoint. Checkpoint. It acts as a checkpoint for the anything which needs to enter inside the vascular bundles. This is the checkpoint here. This is a checkpoint here. So what we learn, students? What we learn from endodermis? It is a part of cortex, single layer, intercellular space is absent, made up of parenchyma. It has subarene depositions on the barrel-shaped cells, that is Casparian strips, act as a checkpoint. Any doubt in endodermis now? Any doubt in endodermis? Tell me. Tell me. Any doubt in endodermis? Now this subarene deposition will make sure that water, it is a checkpoint for water actually. Along with water, nothing should enter inside, right? Water is impermeable. Water becomes impermeable. Water becomes impermeable. Done. That is your endodermis. Now, you look at the diagram and tell me now. Look at the diagram and tell me which is the next layer. Cortex done, epidermis done, endodermis done. Inner to the endodermis, do you find which layer now? Pericycle. We find the pericycle. Now, pericycle, if you look at the diagram now, just look at the diagram. Can you see the pericycle? Do you think it's a single layer or multi-layer or few layer? See, this is pericycle, this is also pericycle. Here it's starting, it is also here. That is, can I tell it's a few layer in nature? Yes, it is few layer pericycle. Can I tell it is a pericycle? Can I tell it is few layer? Yes, it is few layer. Then few layer. And can I tell, obviously you should know this, pericycle is also made up of parenchyma here. Parenchyma? No. If you remember in the morphology class, I told you that pericycle is the site for release of lateral roots. Release of lateral roots. See, this is the main taproot. This is the main taproot. From the main taproot, we see some form of lateral roots coming out. Lateral roots coming out from the main taproot. Main taproot. Those lateral roots are originating from, not from the epidermis. They are not originating from the epidermis. Lateral roots are originating from the pericycle, from inside. From inside here, we have the roots coming out. See, this is a section of root, right? This is a section of root. On the epidermis, we have the root hair. Root hair is present on the epidermis. But from the pericycle region, from the inside, lateral roots will arise. From the inside, lateral roots will arise. This is the main root section. From the inside, lateral roots will come out. So lateral roots are arising. And these lateral roots are endogenous in origin. They're what? Endogenous. Endogenous in origin. Because they're originating from the inside the pericycle. From inside pericycle, they're coming out. Lateral roots. That is your pericycle. That is your pericycle. Now students, one more thing you need to remember. I know it's removed from your syllabus, but it is important to understand that your pericycle makes up 1/3 of the vascular cambium. 1/3 of the vascular cambium. That is, it is involved in the secondary growth. Involved in the secondary growth. That's what you should remember. Apart from that, what is secondary growth? How does it play a role, pericycle? How it is dividing? All that is not in your syllabus right now. You don't have to worry even a single thing about it. So only thing you need to know here is that it is involved in the secondary growth, pericycle. Two main functions. First main function is lateral roots. Second main function is secondary growth. Clear? Is that point clear to all of you? Tell me yes or no in the chat now. Quickly, go. This is how you study anatomy, students. This is how you study anatomy. You don't have to, by heart any of this. You need to know from diagram itself, everything should be visible in your head. Pericycle. Done. Now students, tell me, what do you find inside the pericycle now? What do you find inside the pericycle in the chat quickly? Inside the pericycle, do you find the vascular bundles now? Yes. Time for vascular bundles now. Vascular bundles. Now students, if you know, it's a root. It's a sectioning of root. Tell me how the vascular bundles will be arranged. Will they be arranged in the same radii or different radii in an alternating fashion? Xylem, phloem. Which type of arrangement? Obviously, I told you before. In the case of root, it is always radial arrangement. It is always radial arrangement. That is, xylem and phloem separately. Xylem and phloem will be present on different radii. Xylem and phloem will be present on different radii. Now, here if you see, look at the vascular bundles, they are in a patchy condition. Like they are present patch everywhere. Okay. Now, same vascular bundles, what about the location of protoxylem and metaxylem? Look here. If you look closely, this is your protoxylem. Okay, here. Metaxylem is here, somewhere here. If protoxylem, see, metaxylem is here somewhere, protoxylem is towards outside. If protoxylem is towards outside periphery, can I call it exarch condition in the case of root? And metaxylem is towards inside the pith. This is the pith. This is the pith. Protoxylem is towards periphery. If protoxylem is towards periphery, it's called as exarch condition. Yes, exarch condition. Exarch condition. Now, if you look at the diagram here, how many vascular bundles do you see? 1, 2, 3, 4. 4 vascular bundles. So typically, in the case of root system, there are around just 2 to 4 vascular bundles. It's called as diarch or tetrarch condition. Diarch or tetrarch condition. Diarch or tetrarch condition. Done. That's all is vascular bundles. That's all is vascular bundles. Any doubt in vascular bundles? Tell me. Radial, patchy, exarch, just 2 to 4 vascular bundles. Just 2 to 4 vascular bundles. So if pericycle is 1/3 of vascular cambium, how it can contain vascular bundles? How it can contain vascular bundles? It will have vascular bundles, Aparna. It will have vascular bundles. I'll show you. I'll show you NCERT. It'll see. Let me tell you. In the case of stem, what happens is secondary growth. Cambium is present in between xylem and phloem. Cambium is present between xylem and phloem. Towards inside, secondary xylem will produce. Towards outside, secondary phloem will be produced. In the case of stem, but in the case of root, it is radial arrangement. In the case of root, it is radial arrangement, right? Radial arrangement, and you do not find cambium between xylem and phloem here. So technically, even the roots are actually closed. They are actually closed here. But even though they are closed, do they show secondary growth? Yes, they show secondary growth. And one of the main reasons they show secondary growth is because of pericycle also. But how does it play a role? All that is not in your syllabus anymore, right? That was all part of secondary growth. Okay, that was all part of secondary growth. So vascular bundle, no. Vascular bundles are basically xylem and phloem. Vascular bundles are xylem and phloem. Vascular cambium is different. Cambium is different. Okay. Exactly. Cambium will be present somewhere here. It is present between them. You see, here we have medullary rays, right? We have medullary rays. The medullary rays will get converted to cambium later on. Medullary rays will get converted to cambium later on. That is part of your secondary growth. So don't worry about all that now. It's not in your syllabus at all. It's gone. They kicked it out. Okay, they kicked it out. But right now you should know is vascular bundles are here. Vascular bundles are present in a radial arrangement. Xylem and phloem are in an alternating manner here. Patchy, exarch condition. What is exarch? Protoxylems towards the periphery. 2 to 4 vascular bundles. Just 2 to 4 vascular bundles. That's it. That's it. Okay. Now, what about the pith here? You see pith. Tell me. Look at the diagram and tell me. How is the pith here? Students, here it may look large here, but pith is very tiny here. The pith here is very tiny. Pith is very small and also inconspicuous. Also inconspicuous. It is going to be inconspicuous here. Okay, inconspicuous. Inconspicuous. That is, pith is not visible only. Now tell me what is away from the pith now. What do we have? I told you we have steel here. Now tell me in the chat what is steel. What is basically steel? Tell me. What is steel? Steel is basically, tell me, steel is basically pericycle plus vascular bundles plus the pith. Plus the pith. All together they make up the steel. All together they make up the steel. Done? Yes. Now here, technically students, instead of medullary rays, in the case of root, between xylem and phloem, we have something called as conjunctive tissue. We have conjunctive tissue here. Conjunctive tissue is also present here. Conjunctive tissue is also present here. Sir, you said endodermis is the innermost, but pith is the innermost. I told you endodermis is the innermost region of cortex. It is innermost region of cortex. Understand that. See, this is cortex. The innermost, as in the last part of the cortex, is endodermis, part of cortex. Okay. Then we have the pericycle. When we have the pericycle, then we have the pith, then we have the vascular bundles. The entire thing together is called as steel. It is called as steel. Okay, steel. Pericycle plus vascular bundles plus pith. Okay. Understood? The innermost layer of the cortex is called as endodermis. Read the lines. Okay. The outermost layer is called as epiblemma. That is the other name. Epiblemma is the other name of epidermis here. Okay. The cells of the epiblemma protrude in the form of a root hair. That is the appendages. Done. The cortex consists of thin-walled parenchyma cells. Yes, we have cortex is made of parenchyma with some amount of interstellar space. In cortex we wrote down, yes, we wrote down in cortex. Okay. The innermost layer of cortex is called as endodermis, written here again. Endodermis is single layer of the barrel-shaped cells without any interstellar space, I told you. In endodermis, barrel-shaped cells, no interstellar space. Done. The tangentially as well as radial walls of the endodermal cells have depositions of suberin. Yes, suberin depositions as well. Water impermeable waxy material suberin to form the Casparian strips. Suberin depositions there to make the Casparian strips. Done. Next to endodermis is the few layer. See, I told you it is a few layer. Which one? Pericycle is few layer. Diagram of thick-walled parenchymal cells referred to as pericycle. So pericycle is also parenchyma, but it is much more thicker in nature and multicellular. So many layers. Many layer. Now, function of pericycle is initiation of lateral roots and vascular cambium during the secondary growth takes place in the cells. Told you. So pericycle's first function is lateral roots. Second function is to take part in the secondary growth. Right? It is initiation lateral roots and vascular cambium during the secondary growth. The pith is the small and inconspicuous. It means it is not visible at all. Pith is not visible at all here. Okay. The growth takes place in these cells. Okay. Parenchyma cells. The parenchyma cells which lie between the xylem and phloem is called as conjunctive tissues. I told you. Between xylem and phloem there is conjunctive tissues present. Conjunctive tissues present. There are usually 2 to 4 xylem bundles, vascular bundles, 2 to 4, diarch and tetrarch condition. We discussed that. It is patchy. Yes. Cambium ring develops between the xylem and phloem later on. That is part of secondary growth. Between xylem and phloem there will be cambium ring. Basically, presence of interfascicular cambium will happen later on. All tissues on the inner side of the endodermis, everything to the inner side of endodermis, such as pericycle plus vascular bundles plus pith is called as stele. It is called as stele. Any doubt here, quickly ask me. Students, do you realize we completely decoded the entire dicot stem with the help of points and the diagram? And the next time when you're reading NCERT, you don't have to read entire thing. All you need to do is open my notes, read the notes once, and every single point will be clear to you. That's how you understand. That's how you understand how you relate from the diagram you learn. Okay? Now students, with that being said, can we do the monocot root now? Monocot root. Monocot root is very simple. If you finish dicot root, monocot root becomes very easy. Right? Any doubt, quickly ask me. It's getting very cold here. Once again, students, any doubt, quickly ask me in the chat. No doubt? Okay. If there is no doubt, can we go? Can we start? Yes. Students, in the case of monocot root, let me tell you, the epidermis, cortex as well as the endodermis is almost same. Okay. In the pericycle. Obviously, pericycle will cortex. When I say cortex, obviously endodermis is included. The epidermis. Okay, why is it so thick? Epidermis cortex and pericycle as well as pericycle is almost same to dicot root. Monocot root, dicot root, everything is same. Now what is the difference then? What is the difference? The main difference will come in vascular bundles. Main difference will come in vascular bundles. Main difference comes here. For example, you look at the number of vascular bundles now. First of all, look at the number of vascular bundles. In the case of dicot, it was 2 to 4. You remember 2 to 4 vascular bundles. But in the case of your monocot route, count 1, 2, 3, 4, 5, 6, 7, 8. 8 number of vascular bundles are here. Yes. That is basically your xylem here, but phloem will be here somewhere. Phloem also will be there somewhere. Yes. So it's also called as polyarc condition. Polyarc condition. That is more than 4. Is usually polyarc condition. Yes, polyarc condition. But if you look at the vascular bundles again, this is monocot route. Monocot. In monocot, do you see secondary growth? Obviously, sir, the secondary growth is absent here. Absent. So can I call the vascular bundles here closed vascular bundles? Can I call them? They are closed. Vascular bundles are completely closed here. Yes, they are closed here. Okay, the vascular bundles are completely closed here. Apart from that, if you look at the diagram, all of you know, apart from that, it's a route. It's a route. So tell me, how will the vascular bundles be arranged? If it is a route, vascular bundles will be arranged in radial fashion. Radial and X arch will be the same to same because it's a route. Since it's a route, radial and X arch, that is protoxylem towards the periphery, X arch condition. Done. That is your vascular bundles. Now, apart from that, the major difference is going to come in the pith here. Can you see this pith? The middle part. The pith here is comparatively large. The pith here is comparatively large here. The pith is going to be large here. The pith here is going to be comparatively large as well as well developed. It is well developed. Well developed here. Done. That's all. It's done. Monocot route is done. Everything else is very similar to it. Everything else is very similar to dicot. If you can read here, it says that the anatomy of monocot route is similar to the dicot route in many aspects or respects here. Okay. It has epidermis, cortex and endodermis and pericycle, vascular bundles and pith. Everything is same to same. As compared to the dicot route, which have a fewer xylem bundles. That was the first difference. Xylem bundles were changed, number was less. Right? In the case of dicot, xylem bundles were comparatively less in dicot. But in the case of monocot, it is polyarc condition. There are usually more than 6 polyarc xylem bundles in the monocot route. So in the case of monocot route, it is polyarc condition. Okay. Now, pith is very large and well developed. Monocot route do not undergo secondary growth. If they do not undergo secondary growth, obviously it is a closed structure. That is, between xylem and phloem, there is no type of cambium. Vascular cambium is completely absent here. It is closed. Done, students, done. Monocot route is done. Look at the question now. Students, is the question visible to you? Is the question visible? Let me read it for you. Is the question visible? Tell me. Yes or no in the chat quickly. Friends, the transverse section of monocot route shows the following internal tissue organization. Arrange them in correct sequence starting from the periphery to the center. I think it's periphery only, right? Starting from the periphery towards the center. So here they have given you, students, different types of structures, You need to arrange them in a order. What is the order? From periphery to the center, how are they arranged? Periphery to the center, how are they arranged basically? This is the maximum zoom I can do. I can do more, huh? Okay. Visible now? I know it is little sota, but no. This is the best I can do. Now, see this. First is the endodermis, pith, then epidermis, pericycle, cortex. Tell me, which is the outermost layer? outermost layer is going to be the epidermis. After epidermis, we have the cortex. After cortex, the innermost layer of cortex. The innermost layer of cortex is endodermis. After that, we have the pericycle. Then we lastly have the pith. Yes. So answer is option number C, E, A, D, E, right? B is the last. Yes. 3 is the answer. Look at this question now. Look at the question. It's all NEET PYQ, students. All NEET PYQ. The transverse section of plant part shows polyarch condition, radial arrangement, and exarch xylem with endodermis and pericycle. The plant part is identified to be. They're asking you to identify the plant part now. Okay. Now, first of all, as soon as you see the exarch condition as well as radial condition, you should be able to tell, sir, it is a root. So can I remove the stems now? Stem is gone. Now, in dicot root, you saw 2 to 4 xylem bundles. But this polyarch condition is found in the case of monocot. So 1 is the answer. Easy. 1 is the answer. Now look at this question. Look at the question. It says that initiation of lateral roots and vascular cambium during secondary growth takes place in the cells of. Time starts now. I'll give you 5 seconds. Go. Try answering this question. Lateral roots as well as vascular cambium is produced in. This is the NEET 2022 question. 2 years back question. Students, do you realize how the questions will be asked? Based on the points which we learned in today's class, question can be crafted really well and given to you. Okay. Obviously, it's going to be the pericycle. Look at this question now. In dicot root, the vascular cambium originates from. Where do you get the vascular cambium in dicot root? Tissue located below the phloem bundle and a portion of pericycle tissue above the protoxylem. Cortical region. Parenchyma between endodermis and pericycle. Interfascicular and interfascicular tissue in a ring. Student, this was one of the controversial questions. Darshini, don't worry, I will take. Don't worry. I will take. I'm here only. I will take. Yes. Yes, Ravindrani, option number A is correct. Tissue located below the phloem bundles. See, below the phloem bundles, we have below the phloem bundles and a portion of the pericycle. See, a 1/3 of pericycle, I told you. 1/3 of pericycle and rest of the tissue located near the phloem. Okay. It is not going to be parenchyma between endodermis. No. Students, such questions will not be asked to you anymore because it is the part of your secondary growth. Such question will not be asked to you right now. So carefully. So I told you this, thought you this part. That's why I'm asking you this question. Okay. Go. Time starts now. Casparin strips occur in. Where do you find Casparin strips? Epidermis, pericycle, cortex, or endodermis? Go. Time starts now. Where do you find Casparin strips? Students, if you are new to the channel, if you are watching the stream for the first time, quickly smash that like button and share the video. Because if you want to solve such questions right, you need to know every line of NCERT, you need to remember the diagram for it, right? Casparin strip, that is barrel-shaped cells with the suberin deposition. Where did we notice that? Barrel-shaped cells with suberin deposition. We saw that in endodermis. Yes, we saw that in endodermis. None. Now students, tell me, did the questions help you? Now? Yes, it helped you, right? So that is what I am doing. So after every concept, we have questions. So we'll deal the questions one by one and we'll go further ahead. Okay. The next topic is going to be your dicot stem. Let me know in the chat if you're ready for dicot stem now. If you're ready for dicot stem. Okay. Dicot stem. Now. Can we start? Can we start, students? Quickly let me know in the chat if we can start the dicot stem now. One by one we will decode the entire diagram. Nothing to be by hearted here, nothing to by heart. Just look at the diagram, write down the information, that's it. You do not have to even by heart anything here. Okay? Can we start? Yes. Now, first of all, tell me which is the outermost layer here. Which is the outermost layer? Outermost layer is going to be epidermis here. Obviously, students, the outermost layer is going to be epidermis. Okay? Epidermis is the outermost layer here. Now, tell me, if you look at the epidermis, does it have any epidermal hair here or epidermal appendage? Yes. Can you see some epidermal appendages? Yes. That is basically trichome. Yes. So can I just call it as epidermal hair right now? Epidermal hair. Yes, epidermal hair is present here. Now, just look at the epidermis, students. Just look at the epidermis. Do you think there is any type of intercellular space here? Yes. Do you find any type of intercellular space here? Here? No. Intercellular space is mostly absent here. Yes. Now, what about cuticle? What about cuticle here? So, students, in cuticle, in the case of some type of, in the cuticle will be present in some stems. It is not present in some stems. So, can I tell cuticle may be present or may be absent? Yes. Cuticle may be present or may be absent. That is cuticle. Okay. Now, what about stomata? Stomata. Again, stomata can be present or cannot be present in many cases. It is present or it may be present or it may not be present. It may be present or may not be present. Okay. Now, if you look at the epidermis, how many layers do you think it is? How many layers? Obviously, it is single layer. Single layer. And it is made up of parenchyma. It is made up of parenchyma. It is made up of parenchyma. Done. Students, do you realize epidermis, all the points is here just from the diagram. If you look at the diagram, all these points should be in your head. That is, epidermal hair is present, intercellular space is absent, a single layer made up of parenchyma, cuticle may be present, may not be present, stomata may be present or may not be present. Done. Epidermis is done. Now, students, let's look at the cortex now. Let's look at the cortex. Let's look at the cortex. In the cortex, here in the case of stem, the cortex is divided into three parts. The cortex is divided into three parts. Cortex is divided into three parts. Okay. The outermost layer of the cortex. The outermost layer of cortex right here in the diagram, try to find it. Which is the outermost layer here? Outermost layer in the cortex. Obviously, if this is the entire cortex here, this is the entire cortex, from here to here is the entire cortex. The outermost layer, can you see this one? The outermost layer is right here. Yes, the outermost layer is called as hypodermis here. Cortex first layer is called as hypodermis. Hypodermis is the outermost layer. Hypodermis is made up of, remember students, when I was teaching you about 𝐂𝐡𝐞𝐤, hypodermis in the case of dicot stem is made up of 𝐂𝐡𝐞𝐤. It is made up of 𝐂𝐡𝐞𝐤. Made up of 𝐂𝐡𝐞𝐤. That is your hypodermis. Now, in the hypodermis, do you find again any type of intercellular spaces anywhere? Just look at the diagram and tell me. Tell me. Obvious, students, there is no intercellular space here. So, can I write hypodermis, intercellular space is completely absent. What could be the function of hypodermis, students? What could be the function of hypodermis? So, if you remember, in the case of stem, a stem is very flexible. Yes. As a petiole is very flexible. Yes. So, main function of 𝐂𝐡𝐞𝐤 right here is to provide mechanical support. The main function here is mechanical support. Mechanical support. That is basically, when I'm trying to tell mechanical support, it is flexibility. Flexibility. Okay, it is going to be flexibility is the answer. That's it. That's it. Hypodermis is done. Do you see anything easy? So easy. Tell me the second layer. The second layer is cortical layer. Is the cortical layer here. Second layer is going to be the cortical layer. Now students, in the cortical layer here, the cortical layer, it is made up of parenchyma. First of all, it is made up of parenchyma here. Parenchyma. It also has some form of intercellular space is present here. Can you see? Some form of intercellular space is present here. Some form of intercellular space is present here. Yes, it is present, students. Now, apart from that, in your cortical layer, it is not just a single layer. Can I say it's a few layer also? It is few layers. It is few layers. It is going to be few layers. That is going to be your cortical layer. Second one. Now tell me, which is the innermost layer of your cortex? See, first layer is hypodermis here, collenchyma. Second layer is parenchymatous cortical layer. The innermost layer, can you see this one? The innermost layer of your cortex is technically your endodermis. Can you see this here? This is the innermost layer. That is endodermis. The third layer and the final layer is going to be endodermis. Endodermis is going to be the innermost layer. You see, endodermis is the innermost layer here. Endodermis. Now students, in the case of your dicot stem, the endodermis is very special. Endodermis is very special here because the endodermis has something called as starch sheath here. It has something called as starch sheath. Has something called as starch sheath. Starch sheath in the endodermis. Done. Next layer. Now look at the diagram and tell me. Look at the diagram and tell me which is the next layer to endodermis. The next layer to the endodermis is nothing but the pericycle here. Can you see? And if you look at this diagram right here, can you see the pericycle is kind of wavy here? Can you see? The pericycle is kind of wavy. Yes. The next layer is going to be pericycle. Pericycle. Pericycle right here is little wave-like. Wave-like. Yes. And if you look at the shape of the pericycle here, look at this. Look at this shape of the pericycle. This is full moon, student. This is full moon. This is half moon. This is called as half moon. Does it look like half moon? Yes. So can I tell it is semi-lunar? It is semi-lunar in shape. Can you see? It is semi-lunar in shape. Yes. And the most important part here is the pericycle is made up of sclerenchyma. It is made up of sclerenchyma. That is your pericycle. 3 points done. 3 points done about your pericycle. Any doubt in pericycle? Tell me. Any doubt in pericycle? No. The next thing, if you look at after pericycle, we have the arrangement of vascular bundles. Yes. We have next thing is the vascular bundles here. First main point about vascular bundles is the arrangement of bundles here. Arrangement of bundles. Arrangement of bundles, if you look here, can I tell the arrangement of bundles is something like a ring arrangement? Ring arrangement of vascular bundles? Yes. It is ring arrangement. Ring arrangement here. Ring arrangement. Yes, it is ring arrangement. Now, since it is a stem, will the vascular bundles arranged in different radii or same radii? Can you see? Vascular bundles are arranged in the same radii, phloem, xylem, phloem, xylem, phloem, xylem, and that is called as your conjoint arrangement. It is called as your conjoint arrangement. Yes. And if tell me in the chat, in the vascular bundle, if xylem is towards the pith and phloem is towards outside, the xylem is towards the pith, what do we call it? When the xylem is towards the pith, we call it collateral. Yes. Collateral arrangement. Here, done collateral. Now, since it is a stem, right? What will be location of protoplasm here? Look at the protoplasm. Here, look at the diagram. Don't believe me, look at the diagram here. Protoxylem is towards the pith now. You see, this is the protoxylem, this is the pith. If protoxylem is towards the pith, what do we call that? When the protoxylem is towards the pith, we call it endark arrangement. See, if protoxylem is towards periphery, we call that exarch. If protoxylem is towards the pith, we call it endark arrangement. Endark. Yes, we call it endark arrangement. Done. Now, apart from that, between the vascular bundles, we find, where is it? It's not given here. Huh? Cambium is present. Between the vascular bundles, we find cambium here. Yes, it's a dicot stem. In the dicot stem, between the xylem and phloem, we find vascular cambium. So, is it open or closed? Tell me. It's going to be open. It's going to be open. Done. Those are all the points when it comes to your vascular bundles. Done. Done. Now, students, between the vascular bundles now, can you see here? Between the vascular bundles, we have basically the medullary race. Between the xylem and phloem, between the vascular bundles, we have the medullary race, which is made up of parenchyma. Medullary race between the parenchyma. Done. Any doubt here? No, nothing. Cambium is present here. Can you see here also is given here? Cambium is present between xylem and phloem. They are present in same radii. That is basically your conjoint. Now the only thing which is left is pith. Now let's do pith also. Now look at the pith. What can you tell? If you just look at the pith, what can you tell, students? Yes, the pith is very large here. Yes, pith is very large. What is it made up of? It is made of parenchyma. Yes, the pith is very large. Made up of parenchyma. It may not be visible here because in the case of stem, the pith actually, pith actually has some type of intercellular spaces is present here. Intercellular space is present in the case of pith. Done, students. Done. With that, pith is also covered. Now. So where do we start from? We start from the epidermis. Epidermal hair, no intercellular space here, cuticles may not be present, stomata may or may not be present, single layer parenchyma. Then we came to the cortex. Cortex, 3 layers. First is hypodermis, made up of collenchyma, intercellular space is absent, mechanical support, flexibility. The second layer is called as cortical layer, parenchyma, intercellular space present, few layer thick. Then there we have the third layer, endodermis, which has starch sheet. Then we have pericycle, wave-like structure, semilunar, sclerenchymatous. Then we have the vascular bundles, ring arrangement, conjoint, collateral, endark, and open. Then we have the pith, large parenchyma, intercellular space is present. So did you realize one by one? Look how it looks like. You just go one by one. I think similar thing which we did for even similar way we did for your dicot root also, right? Yes. Students, when you're studying, this is how it should look like. Separate page. Epidermis, cortex, endodermis, then pericycle, then we had the vascular bundles, and finally we had the pith. Then we read the NCERT. Similarly, we did for your entire dicot stem. See here, dicot stem. Epidermis done. Cortex, pericycle, vascular bundles, pith, and done, and finished, and done. So that's how you need to learn. That's how you need to learn the entire taxonomy, entire your anatomy, okay. Now see here, let's read the lines of NCERT. What does it say? That the transfer section of your typical young dicot stem shows epidermis, outermost productive layer, yes, of the stem, covered with thin layer of cuticle. I told you cuticle is present on epidermis, yes. It can be trichomes, basically, which is the stem here, yes, and have few stomata, yes, stomata also present, I told you. Multiple layers below the epidermis is called as what? Hypodermis. Very context, it is a consist of 3 zones. The outer is called as hypodermis. Hypodermis is made up of collenchyma. Just below the epidermis with productive mechanical strength. See, functional mechanical strength to the young stem. Cortical layer is the second one. First is hypodermis, cortical layer is the second one of the cortex. Thin walled parenchyma cells, yes. Intercellular space is present, I told you. The third layer is called as endodermis, third layer of cortex. Okay, they have something called as starch sheet is present in the case of endodermis. What about pericycle? Where is it? Pericycle. Pericycle is semilunar, made up of sclerenchyma. Yes, it is wave-like. I told you, wave-like. Vascular bundles are there. Yes, arranged in a ring form. We did that ring form arrangement. We did. Then conjoined, open, endarc, protoxylem. All the points one by one we covered. Yes. Then we have a large number of rounded parenchyma cells with a large intercellular space occupies the central portion of the stem called as the pith. That is the last part I told you. Pith we wrote here, right? Pith. Large parenchyma, intercellular space. Pericycle done, everything is done. Dicot stem is over. With that, dicot stem is over, friends. Any doubt in dicot stem? Tell me now. Any doubt in dicot stem? I believe there should be no doubt. Now that was your dicot stem. Now monocot stem is somewhat similar, also different at the same time. Let's deal with monocot stem, then we'll take a small break. Okay, let's deal with the monocot stem. Very easy again, very easy, nothing to worry or anything. It's even more easier. Okay, now if you look at the monocot stem, first of all tell me which is the outermost layer again here. Which is the outermost layer? Just look at the thing and tell me which is the outermost layer here in the monocot stem. The outermost layer is going to be epidermis. Epidermis is similar to your dicot. Epidermis is same. Epidermis is same to same, students. Epidermis is same to same when it comes to monocot and dicot, same to same. But the major difference is coming in hypodermis now. Major difference will come in hypodermis. Now, haven't I told you, collenchyma is absent in the case of monocots. Collenchyma is completely absent in the case of monocots. So, what do you think the hypodermis is made up of then? If collenchyma is absent, your entire hypodermis right here, entire hypodermis is made up of sclerenchyma. It is made up of sclerenchyma. Made up of sclerenchyma. Done. Okay, that was epidermis, hypodermis done. Now what is after hypodermis? Tell me. After hypodermis, what do we get? After hypodermis, we have the entire cortex. Now cortex here is little what is the meaning of that? It is not very differentiated. The cortex here, that is the ground tissue. The entire ground tissue, can you see, this is the entire ground tissue. Entire ground tissue is not differentiated. Entire ground tissue is not differentiated. It is just made up of parenchyma. Just remember that much. Entire ground tissue is not differentiated. Not differentiated. And it is made up of parenchyma. It is made up of parenchyma. That's all you should know. Okay, done. Now what about vascular bundles, students? What about vascular bundles? I think we should have added one more slide here, but okay. We'll write here, students. We'll write here. I can't copy this image, huh? It's fine. Okay, let's not copy the image, let's just do it from here only. Tell me, what about the vascular bundles? Do you think they're arranged in any specific structure here? Do you think the vascular bundles here, do you think they're very properly arranged somewhere? No. So this point is very important. This point is very important. It's the identification feature for your monocot stem. The identification feature for monocot stem is scattered vascular bundles. That is the clue. Write down for your entire structure of your vascular bundles. In the case of your monocot stem is going to be scattered vascular bundles. Scattered vascular bundles. Now, if you look at the diagram, I want you to focus very carefully now. Listen to me very carefully now. If you can see here, the outer vascular bundles, they are very tiny. Outside they are very tiny, but they are very large in number, many in number. But as we go towards the center, the size of the vascular bundles towards the center is much more larger. Yes, it is much more larger, but it is very few in number. Very few in number. Understand that. That is, vascular bundles towards the outside are many, but small size. But they are very small size. But same vascular bundles towards inside are few in number. The few in number, but they are very large size vascular bundles. Done. Last one. Now, one of the most important point about your flow, about your monocot stem, is that the phloem parenchyma is absent here. Very important point. The phloem parenchyma is absent. Phloem parenchyma is going to be absent in the case of monocot stem. Now, apart from that, if you look at the vascular bundles, since it is a stem, it is a monocot, do you think they are open or closed? Tell me, do you think they are open or closed bundles here? Obviously, if it is a monocot, they are going to be closed. But it is a stem, so it will be conjoined. They will be present together, conjoined and closed. Yes, conjoined and closed. And if you look carefully, students, at the vascular bundles, in the middle, do you see this blue color structure here in the middle? And if you look at your NCERT also, there is some form of blue structure within the vascular bundles. The blue structure within the vascular bundles is called as water cavities. It is called as water cavities. Okay, so water cavity inside vascular bundles. Water cavities is present. Water cavities is present. Done. That was the entire monocot stem. Let's read the NCERT. See here, it's very simple. Monocot stem has a sclerenchymatous hypodermis, I told you. A large number of vascular bundles, each surrounded by a sclerenchymatous bundle sheet. Okay, we didn't write that. Let's write down. Each vascular bundle is covered by a bundle sheet. Each vascular bundle is covered by bundle sheet, which is made up of sclerenchyma. Here, made up of sclerenchyma and a large conspicuous parenchymatous ground tissue, scattered vascular bundles. Vascular bundles are conjoined and closed. All of you know that. the peripheral vascular bundles are generally small. On the outside are generally smaller, but than the centrally located ones. Okay. The phloem parenchyma is absent here. Very important point. Water containing cavities are present within the vascular bundles. The small blue things you saw present inside the vascular bundles. Done. We'll do some questions, then we'll take a small break, then we'll do the leaf, and then we'll do questions more. And at the end we have even more questions. Okay. Look at this question now. Go. Time starts now. Look at this question. A major characteristics of monocot root is the presence of. In monocot root, what should be the characteristics here? Do they have open vascular bundles? Tell me, yes or no? Do they have open vascular bundles? No, they are closed. Scattered vascular bundles? Yes. This is also correct. Vascular bundles are scattered here. Vasculature without cambium? This is also correct. Cambium sandwiched between phloem and xylem? Cambium sandwiched between phloem and xylem along the radii? They are not sandwiched here. Okay. So if you between 2 and 3, tell me which is the most probable answer now? Which is the most probable answer? So if you look at your NCERT, under your monocot stem, has they ever mentioned the scattered vascular bundles anywhere? Look. Even though we write and we learn in diagram, look at the NCERT. Has they ever mentioned anywhere? Have they ever mentioned? No, they have not mentioned. They have not mentioned anywhere. So technically, students, most probable answer is going to be without vascular bundles. That is basically closed condition. The closed conditions. So you will get such tricky questions sometimes. So you need to think ahead and look and remember lines of NCERT. Okay. Next question. Interfascicular cambium develops from the cells of. Okay, I think I mentioned this. Interfascicular cambium develops from the cells of your medullary race. From the medullary race. Medullary race gets de-differentiated to interfascicular cambium. Interfascicular cambium. Water containing cavities in vascular bundles are found in. Where do you find water containing cavities? It was root, I didn't check that. Oh, it was root then. Okay. So if it is root, obviously it will not be scattered because it is in a ring arrangement. It will be in a ring arrangement. This is the answer. That's all. Easy then. Then even more easy. Where were we? This was done. Look at this answer. Water containing cavities in the vascular bundles are found in. In vascular bundles they are found in sunflower, maize, cycas, pinus. Students remember, cycas and pinus are gymnosperms, so this is gone. Between sunflower and maize, tell me. Sunflower and maize. Between sunflower and maize, which one is part of your monocot, which is dicot? Obviously, sunflower belongs to Asteraceae, which is dicot, and your maize is a monocot, grasses family. Yes. So, you know, in the case of your monocot stem, water cavities are there. Then this is the answer. Maize is the answer. Yes, maize is the answer. So with that, your entire stem of monocot and dicot over. Root, monocot and dicot both is over. Now we'll take a 15 minutes or 10 minutes break and we'll come out. Okay, we'll take a 10 minutes break and we'll come back for the leaf. See here, we have the leaf to do. We have the leaf, monocot leaf, and then we have a lot of questions. We have lots and lots of questions. Can you see? We have so many questions ahead. So we'll be finishing all those questions also today. Okay. So what is the time right now? The time right now is 7:55. So at 8:05 we'll start. 8:05 will start. Okay? 8:05 class will resume. Okay? So let's take a 10 minutes break so I can drink some water and come. And with that, take care all of you. The mic only dropped. The mic only dropped. 1 second. I don't know how to fix the mic now. Done. Done. Students, let me know in the chat if you're ready for the part 2 of the, not part 2 technically, it's almost, the chapter is over. The last part of the chapter, that is the leaf. And leaf is actually technically very easy, and there is nothing much here. There's nothing much to study here. Leaf is very tiny topic. And the major chunk of the chapter is almost over, and you need to actually go to the last part. Done? Let's start now. One thing, let me drop a message. Never mind. Now students, let's quickly draw a leaf now. Let's quickly draw a leaf. Leaf. Yes. Okay, this is your leaf. Then we have veins in the inside. We have a large midrib. We have veins. And we have veinlets. As well as we have the veinlets. Okay. We have veins and veinlets. Now students, the outer layer is called as what? Tell me. What is the outer layer of the leaf called as? The outer layer is called as epidermis, obviously. It's called as, 1 second. The outer layer is called as the epidermis here, right? Okay. Now what about this side? This side is also called as epidermis. Yes. Because the leaf will have two sides. The leaf will have two sides. So both the sides is called as epidermis only. But students, you need to understand here is the epidermis of upper layer and lower layer are completely different. They're completely different. Now in terms of stomata, in terms of cuticle, everything is different. Even the names are different. In fact, the upper layer is called as adaxial. It is called as adaxial, also called as ventral side. It's also called as ventral side. Now what about the lower side? The lower side is called as, let me write lower. Lower side is called as abaxial side. It's called as abaxial, or tell me what is the opposite? Let me drink some water. What is the opposite of ventral? It starts with a D. Dorsal. The lower side is called as dorsal side. Also called as dorsal side. Done. It's called as dorsal side. Now what about in the case of monocot and dicot? Now this is a typical dicot leaf. Yes, it's a dicot leaf. So how can you tell me, sir, this is a dicot leaf? Because look at the arrangement of veins and veinlets. Here the arrangement of veins and veinlets is in the form of a network. So it is a dicot leaf. Now what about if a leaf, imagine we have a leaf like this now. In this leaf, from the midrib, all the veins are parallelly arranged like banana leaf. This is called as parallel venation, and this is a monocot. This is a monocot leaf. This right here, the bigger one, is technically a type of dicot leaf. This is a dicot leaf. Now, there's one simple concept here. There's one simple concept. Let me explain. The simple concept is that in the midrib region, this here is the midrib region. On the midrib region, there are more vascular bundles compared to the other veinlets. Compared to the other veinlets, midrib will have more vascular bundles. But in the case of monocot, the number of vascular bundles, this is more means number. In the case of monocot, same number of vascular bundles, same number of vascular bundles throughout. The number of vascular bundles will be same to same in the case of monocot. But in the case of dicot, the midrib will have more vascular bundles. As we go away from the vein or veinlets, the number of vascular bundles will vary. That's all. Done. Now students, let's go to the dicot leaf. Again, dicot leaf has an upper epidermis and lower epidermis. Upper epidermis is called as adaxial, lower one is called as abaxial. Backside is called as abaxial. Yes, abaxial. And if you look at the leaf, dicot leaf here, if you look at the arrangement of mesophyll cells. The arrangement of mesophyll cells. The mesophyll cells here are divided into two categories. One is called as your palisade mesophyll. The other one is called as spongy mesophyll. So students, in the case of dicot leaf, the mesophyll cells are divided into two categories. One is your palisade, other one is spongy parenchyma. Is that point clear to all of you? Yes. Now, if you look at the palisade, palisade mesophyll, they're arranged parallel to each other. Yes, they're arranged parallel to each other. Yes. Now, what about spongy? Spongy are not parallel, but they're almost in a vowel shape and they're connected to each other with a lot of air gap, a lot of air cavity. And students, if you look at the dicot leaf, if you look at the typical dicot leaf, on the lower side, you will have more number of stomata. In the case of dicot leaf, lower side will have more stomata. But in the case of monocot, number of stomata will be equal. There. Okay. Here more stomata on the abaxial side, more stomata. Now we have something called as bundle sheet here. Bundle sheet. Bundle sheet is basically a covering bit for your xylem and phloem. If this is xylem and phloem is here, it is covered by a sheet around it. Yes, that is called as bundle sheet. And bundle sheet is made up of parenchyma. Now, do you think cuticle will be present? Tell me, students. Do you think we have cuticle here? Yes, cuticle will be present because it is a leaf. Cuticle has to be present. Stomata, yes. Stomata also has to be present. Now, what about the vascular bundles? How are they arranged here? Do you think they are open or closed? Obviously, they will be closed here. And they'll also be conjoined. Done. That's all is the entire dicot leaf. Nothing extra. This is what is given here. Let's read it once and you will understand all the problems will go away in your life. See here. First one you need to know is dicot leaf is called as dorsiventral leaf. A vertical section of dorsiventral leaf through the lamina shows three main parts. That is outer epidermis, mesophyll cells and vascular system. Three main parts: outer epidermis, middle cortex, that is mesophyll cell, and the inner vascular system or vascular bundles. Okay. The epidermis, which covers the both upper surface is called as what? Adaxial surface, and the lower surface is called as abaxial surface. Okay. Has conspicuous cuticle. Obviously, cuticle will be present here. Abaxial epidermis generally bears more stomata. I told you the lower side will have more stomata. It's given here. More stomata. Adaxial. The later may even lack stomata. That is, on the upper surface, stomata also might be absent also, because so much of sunlight. If more sunlight, more transpiration loss. Do the plants need that? No. So in the upper side, sometimes stomata is completely even absent on the upper side. That is adaxial side. The tissue between the upper and lower epidermis is called as mesophyll. And mesophyll is divided into two categories: palisade parenchyma as well as spongy parenchyma. Done. Your mesophyll cell, what is the main function? Photosynthesis. Okay. The adaxially placed. Why adaxial here? Because on the adaxial side we have the palisade parenchyma or palisade mesophyll cell on the adaxial side. Right? Palisade parenchyma is elongated, which are arranged vertically and parallel to each other. So they are elongated. They're elongated cells and they're arranged parallelly to each other. That is palisade parenchyma. Okay. Now, your oval to round shape spongy parenchyma, that is there. Yes, that is situated below the palisade cells and extends to the lower epidermis. There are number of large spaces in the air cavity. There is obviously air cavities present here. Done. Between the vascular system. Includes vascular bundles which can be seen in the veins and midrib. I told you, in the veins and midrib we find the vascular bundles, and in the case of midrib you will find more in number, right? The size of the vascular bundles are dependent on the size of the veins. Bigger the veins, more size. The veins vary in thickness in reticulate venation, that is dicot leaf venation, or the dicot leaf. Vascular bundles are surrounded by a layer of bundle sheet. I told you, bundle sheet, and it is made up of parenchyma. It is made up of parenchyma. Done. Then look at the monocot leaf now. That is dicot leaf, that's all. There's nothing extra here. Monocot leaf is even more simple, students. Monocot leaf is even more simple. Look at the simple monocot leaf. It has adaxial and it has abaxial. Abaxial has stomata again. But if you look at the major difference here, the major difference is going to be there is no separation of palisade and spongy here. Gone. The separation of palisade, spongy, the concept is not present in the case of monocot leaf. You only have your mesophyll here. Only mesophyll. And if you want to notice, in the case of your monocot, because of parallel venation, the number of stomata is equal on the both the side. It is equal on the both the side. See here. The anatomy of isobilateral leaf, it's called as isobilateral first of all. That is your monocot leaf, is similar to dorsiventral. What are the changes here? The stomata are present on both the surfaces, I told you. Both the upper epidermis, lower epidermis. Adaxial, abaxial, the number of stomata is going to remain the same here. Remain the same. And the mesophyll is not differentiated. I told you, mesophyll is not differentiated into spongy and palisade. Only we have mesophyll, there is no differentiation. There is no differentiation. Okay. Now here there is one more concept called as bulliform cells. Bulliform cells are large colorless cells which help in storing water. That's all. The grass is certain on the adaxial epidermis. Along the veins modify themselves. Okay. In grasses, certain adaxial epidermal cells along the veins modify themselves into large, empty, colorless cells. So here we have the epidermal cells, right? In the case of monocots, these epidermal cells, some of the epidermal cells here, can you see? Some of the epidermal cells here modify themselves. They modify themselves to become colorless, right? Colorless, large cells. They're called as bulliform cells. They're called as bulliform cells. Now, what is the main function of bulliform cells? The main function of bulliform cell is obviously to store water. Obviously to store water, but every single time there is no water, what happens? If this is the plant leaf, the plant leaf will start to curve like this. Can you see? The plant leaf will start to curl like this. This is called as curling of leaf. It's called as curling of leaf. And why does it happen? Because if there is water loss, bulliform cells will lose water, the leaf will start to curl. Okay. When the bulliform cells in the leaf have absorbed water, they are turgid. When water is there, it is very turgid. Deep surface is exposed when they are flaccid. Meaning of flaccid is when the water is being lost. Once the water is being lost, all the water is gone. Once the water is lost, the leaf will start to curve like this. That's all. So can you explain about the bundle sheet extensions? Bundle sheet extensions is not in your syllabus. This is the only thing. See, students, the only thing you need to remember is where does the bundle sheet originate from? Bundle sheet. About bundle sheet extensions. Bundle sheet, the only thing you remember about bundle sheet is that you need to know that it is made of parenchyma, nothing there. Trust me, students, they will not ask you more than that. They'll never ask you more than that. Now, in bulliform cells, if the water is being lost, they will start to curl like this. Okay. The leaf curl inwards to maintain the water loss. The parallel venation in the monocot leaves reflects the inner similar size of the vascular bundles. So the size of the vascular bundles is similar throughout in the case of monocot. In the case of dicot, midrib bigger. In the midrib region, much more bigger vascular bundles. I think I told number here, it is supposed to be size. No, it should be size. Size is more, not number. Size is more near the midrib region. Students, with that actually the chapter is over. With that the chapter is over. Now let's solve some questions now. Question time. Let's do some questions. Okay. What is the question now? Big question is here. Let's read the question one by one. In roots, the stem and in root, xylem and phloem in vascular bundles are arranged in a alternating manner. That is called as a radial arrangement. Yes, correct. Conjoined closed vascular bundles do not possess cambium. Yes, if it is closed, no cambium. Okay, correct. In open vascular bundles, cambium is present between xylem and phloem. Yes, it is present between xylem and phloem. It is present. Okay, it is present. The vascular bundles of dicot stem, the third one, dicot stem possess endarch protoxylem. Yes, stem is endarch. Stem is endarch. Root is exarch. Okay, correct. The monocotyledon root, okay, monocot root usually has more than 6 xylem bundles. Yes, it's called as polyarch condition. Polyarch condition is correct. Choose the correct answers from the given options below. All are correct. All are correct. Easy. Okay. Next question. The large empty colorless cells of the adaxial epidermis along the veins of grass are called as bulliform cells. This is a direct question. That was a direct question. It's called as bulliform cells. Bulliform cells. Next question. Bulliform cells are responsible for. What is the main function of bulliform cells? I told you the main function of bulliform cell is to curl inside, right? They do curling, right? Protect the plants from soil stressness, increase the photosynthesis in monocots, provide large space for storage of sugar, inward curling of leaf in monocots. Here. Option number 4. D for doctor, everyone in the chat. Option number 4. Next question. In the transverse section of plant shows the following anatomical features. Students, this type of question is my favorite. They'll give you all the properties and you're supposed to tell which type of stem is it or root is it, right? Large number of scattered vascular bundles surrounded by a bundle sheet. Okay. Large conspicuous parenchyma as ground tissue. Vascular bundles conjoined and closed. See students, if it is conjoined and closed, can I tell it is a type of monocot now? If it is closed, if it is going to be closed, will it be monocot or dicot? Tell me. Conjoined and closed. Conjoined and closed. Tell me. Is it going to be monocot or dicot? Obviously, it's going to be a monocot. Right? It's closed. Right? It is closed. Okay. Phloem parenchyma, I told you this is the most common feature. I think you should have gotten to know by this line here. Scattered vascular bundles, you should have gotten to know. Scattered vascular bundles, you should have gotten to know. It says monocot stem. Monocot stem. This is Now students, I'll ask you a question now. I'll ask you a question. Tell me in which of the following, this is your homework question, okay? Which of the following, monocot root, dicot stem, dicot root and monocot stem, in which of the following endodermis has starch sheet? Which of the following endodermis has starch sheet? Starch sheet. This is your homework question. Homework question in comments. In comments. Students, I've been seeing your comments and it's very nice. It's very nice to see the comments. So I would love to see comments from all of you because the chapter is over. The chapter is over. So Ravindrani, I see all of you. All Ravindrani comment is always there. Priyadarshini, Divya, Tanuja, Meghana, I've seen all your comments. Soni, Madhumati, I've seen your comment. Yes. Hema. With that, your entire chapter is over actually. This is your homework question. Easy question actually. Now students, if you are not a part of DNA batch, I'm telling you this is the offer going on right now also. Let me confirm also one more time. DNA batch right now is at 3,999. DNA plus test series 499, just 1,000 extra. DNA test series plus Tatva book hard copy at 8,000. Students, it doesn't get better than this. It doesn't get better than this. And all of you who are already in DNA batch, who knows it, you know it already. All the students who are in DNA batch, all of you know it, how amazing DNA batch has been, and it's so much of fun. It's so much of fun and everything. So students, let me know in the comment section how was today's class, because anatomy, who will teach you anatomy like this? Tell me. Look at this. Entire structure, every single place we have decoded, right? Every single place we have decoded the entire anatomy. Entire anatomy has been decoded. Who will teach you like this? See? Entire anatomy has been decoded completely. Can you see? Every single diagram by diagram, line to line NCERT with PYQs, every single thing in one place. Every single thing in one place, right? Every single thing in one place. So that was your class by Baswaraj sir, and let me know in the comment section how was today's class, because I enjoyed it. It is personally one of my favorite chapters to teach, because anatomy is all about knowing diagram. If you know the diagram, you're good to go. If you don't know the diagram, it's going to be difficult. So this is my way of learning anatomy. Give it a shot. Try learning the entire anatomy from the diagram only. Then you read NCERT. Okay. So I hope all of you have made notes. If you want these notes, you know where it is available. On Baswaraj Sir Biology Telegram channel. Entire notes will be available to you. And take care all of you. And have a great day. Bye-bye. And have a nice